a new topic for you from the chapter Respiration and Exchange of Gases for class 11, chapter 17. This is the next video. In the previous video, I have discussed about the transport of gases, that is oxygen and carbon dioxide, in various ways. In today's video, I am going to discuss about Bohr's effect versus Haldane effect. So here we go. So student, today's topic is Bohr effect versus Haldane effect. So first I am discussing the Bohr effect. Now in our peripheral tissues, suppose these are peripheral tissues in our body. In the peripheral tissues, the TCA cycle or Krebs cycle is going on as the tissues are metabolically active. So due to the TCA cycle in the tissues, carbon dioxide is released as a byproduct. Now this carbon dioxide, it enters the RBC. Suppose this is the RBC. Okay, let me draw a bigger RBC so that I can accommodate so many things here that I am going to explain. So suppose this is the RBC. You know RBC is a biconcave structure, but I cannot draw that three-dimensional structure on the board. So I'm just trying to make a spherical shape. So this carbon dioxide enters the RBC coming out from the peripheral tissue. Now this carbon dioxide combined with water molecule. In the presence of an enzyme called carbonic anhydrase, for that I am writing Ca, carbonic anhydrase. This carbon dioxide and water produces carbonic acid, H2CO3. That is carbonic acid. Further, in the presence of carbonic anhydrase, this carbonic acid dissociated into proton, that is the H plus ion, and bicarbonate ion, HCO3 minus. Right? So H plus ion and HCO3 minus. This HCO3 minus leaves the RBC and goes in the plasma. This is plasma. So let me write here, it is plasma. So in the plasma, the bicarbonate comes out. Now what happens, as the bicarbonate comes out from the RBC to the plasma, chlorine gets in the RBC from the plasma. So this is what known as chloride shift, and which is otherwise known as Hamburger's effect. So chloride shift takes place. Now what is the fate of this proton? You see, in the RBC there is hemoglobin molecule, right? There is hemoglobin. I am writing Hb for hemoglobin. So this hemoglobin molecule, you see, it is bound with the oxygen. It is bound with oxygen. So where is this RBC coming from? This RBC came from the, I am having this Bohr effect. It came from the lungs. So this is lungs. So from the lungs, the RBC traveled to the peripheral tissue because the peripheral tissue needs oxygen. Now try to understand, this oxygen which is bound to the hemoglobin is there. Now this proton, okay, H plus, it binds with the hemoglobin. As soon as this hemoglobin is bound with the proton, the hemoglobin goes in its tense state. That is the T state. What is the T state? It is a very stabilized state where the hemoglobin is very much, it has no affinity towards oxygen. So why it happens? Because as soon as the proton it get joined with the hemoglobin, the salt bridges, okay, present in the histidine residue of the hemoglobin, it breaks. The salt bridges breaks, and between the dimer 1 and dimer 2 of the histidine residue in the hemoglobin. So what happened? This hemoglobin leaves this oxygen. Oxygen gets dissociated from the hemoglobin because the hemoglobin comes to the tense state, the state in which the oxygen has no affinity to bind with the hemoglobin, and the hemoglobin becomes deoxyhemoglobin as this carbon dioxide. Okay, so here you see this oxygen diffuses out. The oxygen diffuses out from the RBC. Now in the physiological condition, you know, one hemoglobin molecule is bound with 4 molecules of oxygen. Okay, 4 molecules of oxygen it is bound with in normal physiological condition. But here only 2 may leave out from the RBC, but not all the 4. But here, for just to make you understand about the 4 molecule of oxygen is leaving the RBC and coming and binding with the peripheral tissue by diffusion process. Now this HCO3 minus, that is the bicarbonate ion, leaving the RBC, comes in the plasma. Okay, now this bicarbonate ion, what will happen to this, I am telling you. Before that, I may recall one thing that I have already explained in the previous video, that carbon dioxide is transported by 3 methods. What are they? I hope you remember till now. One is as the bicarbonate ion form, that is at around 70 to 75% or 80% as bicarbonate ion form, okay, by the plasma. Next, that is the 7 to 10% of the carbon dioxide is transported in the dissolved state in the plasma because of its high solubility, and nearly 20 to 25% of the carbon dioxide is transported as the carbaminohemoglobin, that is binding of the carbon dioxide with the hemoglobin. So what happens, as this bicarbonate ion leaves the plasma, the chloride ion comes in, which is called chloride shift. The proton molecule binds with the hemoglobin, it brings the hemoglobin to the 10 state, and the oxygen dissociate and enter in the diffuses in the peripheral tissue. Now this RBC is having what? The RBC is having the hemoglobin within it. Let me write RBC. And this RBC is traveling to the lungs. Okay, from the peripheral tissue, the RBC is traveling to the lungs. This hemoglobin, this is bound with the proton and to the carbon dioxide. Clear? Okay, now what happens? In this lung, so it reaches the lungs. In the lungs, this bicarbonate ion, I am putting the dot, that travels through the plasma. Now the venous return takes place in the lungs, the venous blood reaches the lungs. So through the venous return, this bicarbonate ion, it comes to the plasma and enters the RBC. Okay, as soon as the bicarbonate enters the RBC, what happens? The chloride which was present inside, it leaves the RBC and goes out. Here also chloride shift takes place. So here you see bicarbonate enter the RBC. Okay. Now what happened? This bicarbonate, it binds with the proton. Okay. And form carbonic acid again, H2CO3. Now already in the lungs, inhalation has taken place. So inhalation has taken place. During inhalation, you know, oxygen enters in the lungs, alveoli. In the lungs alveoli, suppose this is alveoli, the large number of alveoli. In the alveoli, there is high partial pressure of oxygen. PO2 is high. As the PO2 is high in the alveoli, the oxygen diffuses inside in the RBC. The partial pressure of the oxygen is nearly 90 to 100 millimeter of mercury. So this partial pressure, high partial pressure of oxygen is enough to bind the oxygen with the hemoglobin. The hemoglobin binds with the oxygen and dissociate the carbon dioxide which was bound to it. This carbon dioxide dissociate. And you see this carbonic acid which is formed here by binding the proton with the bicarbonate, it again splits into water and carbon dioxide. Now this carbon dioxide leaves out from the RBC to the alveoli and thereafter from the lungs, the expired air. Through the expired air, this carbon dioxide leaves the RBC. So this process, this is called Haldane effect. Haldane effect. Okay. So I hope you understood now the difference between Bohr effect and Haldane effect. In the Bohr effect, bicarbonate leave from the RBC to the plasma, chloride enters in. The hemoglobin is in the 10 state. Here what happens? Yes, one more thing I need to tell you, that in the Haldane effect, when the oxygen enters the RBC, the binding of the RBC with the hemoglobin brings the hemoglobin in the relaxed state. So the RBC comes in the relaxed state. Why relaxed state? Because the partial pressure of oxygen is high. Again, the salt bridges between the proton and this hemoglobin breaks, and because of that, the RBC comes in the hemoglobin comes in the relaxed state. So here in the Bohr's effect, the chloride is moving in. Here in the Haldane effect, the chloride is moving out. Here the bicarbonate ion is moving out. Here the bicarbonate ion, which is diffused in the plasma, comes out to the plasma and enters in the RBC. So this is all about Bohr's effect and Haldane effect. So hope you understood the entire topic, and if you have any type of queries, do comment me in the comment section below. Thanks for watching the video.